So my privilege today is I have a panel, and I'm going to invite my panelists, Carl, Fyodor, would you please come up here as well? And I have decided to invite Mike Porteus as well. We have online George Church and Peter Marks. Please, let's have a seat. You know, the accomplishments of all the panelists that we have here are so long that if I go through their achievements, we are not going to talk. So I'm going to introduce them briefly, but I think I have to say something. So it's a panel that is quite balanced. So we have 3 physician scientists and 3 patient scientists, because this term was introduced before. We have, again, 2 remote and 4 here. We have what we are disbalanced is in the fact that we have 3 hematologists, I think, and that is probably something that we cannot match. And since the focus today of this panel is really try to think about how medicine would look in the future and anchor ourselves in the present, maybe because we have 3 hematologists, we're going to start there. Obviously, in Hem-Onc today, we are seeing already part of the future. Because, you know, we started the day talking about ALL and AML and myeloma being, you know, not completely cured, but really a fundamental change. So I'm going to start with this particular field, and I'm going to start with you, Carl. As you look at the future, and of course, you all know Carl, right? So I'm not going to introduce him. As you look at the future, at this vision of how medicine is going to look like, what are the signs that you see today in him? And what are the next steps?
Thank you, JC. It's really a privilege to be on this panel. Sure. I mean, I think we're at this, you know, really, I like to say, and, you know, it's Windows 1.0 in this area of cell and gene therapy. I mean, we have now an amazing tool set that we didn't have. And now, you know, we're 5 years after the FDA approval of the first CAR T cell, so first cell and gene therapy. And the good news is, I mean, now there's a half dozen of them, and you know, the response rate has been really unprecedented, and there have been no untoward, you know, unexpected adverse events. I mean, I do worry. We've always worried about insertional oncogenesis, you know, with gene modification. There's lots of workarounds for that, but that has not happened now. When we've added up, Bruce Levine, who's here, the commercials and academic products, about 15,000 patients have been treated now with CAR T cells, and there's not a single case that I'm aware of, at least that's published, of autologous cells where their T cells transforming. So the safety profile is really amazing when you consider, I mean, that's not true, for instance, with stem cells. So at least peripheral blood T cells are very safe if they're autologous. And so that was a big risk. I think we still have other issues to, you know, the other one I was concerned about, and I know they were at the FDA, it was issues like with autoimmunity. You know, we activate cells ex vivo, and would that, you know, by jazzing them up, would that wake up cells that were previously tolerant and maybe lead to autoimmune disease? And if that happens, it's rare. So I think we're at a place now where autologous cells have been a validated platform, and, you know, the exciting issues are this unexpected, to me, you know, the amazing tools that we've seen with genome editing, the ability really to rewrite the genome and make synthetic cells that are more potent and safer. And I think we're going to have, I mean, Matt Porteus, synthetic hematopoiesis. It's unbelievable, the opportunities now, when you consider that the whole playing field includes your bone marrow and all those cells. So I'm very excited about the ability to combine these orthogonal technologies of gene editing and genomic medicine like that with cell therapies.
Do you think that this will be, just to follow up on that note, and then I'm going to go to Peter Marks, do you think that with these tools that we have seen today, and of course we are seeing now, we will move from CD19 for all to more personalized in some cases? There will be a combination of more personal, and what are the implications from the regulatory perspective? Peter, you're going to take that one later, but how do you see that evolution, Carl?
Well, you know, we're at this point where it's an engineering issue of moving, scaling out. It's a scale-out issue with autologous cells, and all the companies are struggling to meet the demand. So I think that's going to be solved, in several ways. In the near term, closed systems and automation, we've heard a lot about that.
Sure.
That has to happen. My big worry is, what if we actually make, engineered cell therapies work for solid cancer? We just couldn't meet the demand at this point. So it's really an emergency to get, a cell manufacturing that's both can be scaled and automated. The process that Novartis is using is what Bruce Levine developed in my lab in 1992. And it's very dependent on FTEs of highly trained people. So that's an engineering issue that's going to be solved. And, then we have these scientific issues now of, how do we go to more complex solid tumors? And I'm very excited that will happen. It's just a matter of when.
Peter, of course, the director of CBER at the FDA, also a hematologist. What, from the regulatory perspective, we heard today actually that we need much more regulatory science going forward, not just at FDA, but in general, in society. And so what have we learned from the genetic medicines and genomic medicines that are already approved, or they are in the process, that might set the stage for all this wave that is coming later?
Yeah, so maybe it's not a popular thing to say, but I think what we've learned is that we have to recalibrate our probabilities of success. For those of us who I worked in industry on small molecules, probability of success for small molecules was not great. You had a small molecule at the beginning, and the likelihood that it was going to ever see the light of day from a preclinical study was pretty small. And it's very different here when we're dealing with these targeted therapies that are targeted through genetic targeting, which is even a next level up from molecularly targeted small molecules. So here, I think we have to prepare for having a reasonable likelihood that you're going to be successful in the end, which means that early on, you need to actually have manufacturing processes that can sustain you through to commercialization. Now, maybe not the first manufacturing process is going to be the commercial process, but I think you can't delay out getting to a process that is commercially viable, because we've seen time and time again products delayed because of this. And toward that end, I think we are hoping to open up dialogue early on with sponsors that are developing products so that we can help facilitate and accelerate that transition. Again, we don't want people to waste money putting together commercial processes for products that are not going to be viable. On the other hand, there's a balance here where we still see all too often products, losing 6 months or a year, or even more sometimes, because they're not prepared for success.
What is the balance? And this is a question for you, Peter, but I'm sure that others will jump at this one. Of course, we see that need that you describe. At the same time, because of this increase in personalization of the therapy, but more a platform for a given class, and Stan talked about it before, we could anticipate, or we could desire, a more platforming approach to regulation, in which there are ASO of certain chemistry is an ASO of certain chemistry, and of course there are risks that are individually based, or the same thing for certain type of editing or something. What are your thoughts and the thoughts of the agency in that direction?
Yeah, so we're very interested in this, and that's one of the reasons why you may be aware that the Foundation for NIH has sponsored the Bespoke Gene Therapy Consortium. The idea here is to try to find ways that we could leverage platforms so that we don't repeat things. You could imagine you have that for gene therapies, you could have a bunch of different inserts in an AAV9. And provided the inserts are within certain certain specifications and for certain uses, ultimately the production would be the same for pretty much all of them. And yes, you'd have to characterize the potency of the different products differently, but by and large, you have a bunch of products that are made the same way, and that might really help us reduce the rework, because you could reference essentially one application for all of these products. But even more importantly, when you make an advance, if you say you had 8 linked products, when you make an advance in manufacturing, all 8 products benefit, instead of what we have now, which is, each time you'd want to go through, you've got to go modify an application for every one of these. And that's why oftentimes, and we see this with CAR T cells, right? People don't want to necessarily advance the manufacturing to the latest technology, because it means going back and investing time, resources, and ultimately money in doing so.
No, that's a fundamental baseline for. I'm going to get Fyodor now. Fyodor, Urnov, UC Berkeley, IGI. Obviously, you are thinking a lot about. You are thinking and doing N of 1 personalized medicines. We had a whole panel about this, so we're not going to go to some of these areas. But you were talking the other day about immune mutations and patients that are affected by it. How do you take Peter's position and apply it now through science to that field, for example?
So first, I never miss an opportunity to thank Dr. Marks for his leadership of the CBER. And I've been injected 4 times, I think, with Moderna and Pfizer. So thank you, Dr. Marks, for helping treat hundreds of millions of patients with vaccines. The other thing to say, it's an extraordinary moment for me as a gene editor, so a relatively new technology on balance, to be in the same room as 2 physician scientists who've enabled entire classes of genomic medicines that have cured thousands of people. So Dr. Stan Crook with Ionis, and of course Spinraza, and Carl June, as mentioned, 20,000 patients treated, 5,000 this year alone. So how do we take a gene editing and, using a specific disease category, for example, inborn errors of immunity, leverage everything Dr. Marks just said and everything we've heard from Danaher and other companies in this space, and build a platform cure? So the first thing to say, Matt Porteus, who was a physician scientist at Stanford, and yours truly met 20 years ago, and when Matt was pioneering gene editing in David Baltimore's lab. And then Carl June did the first gene editing trial, which happened to be editing of immune cells. And the reason I bring this up is we're not at year 1 of gene editing in the clinic. We are at year 14, I think. So there is quite a bit of regulatory precedent and learnings about what we can and cannot do. Matt in his lab at Stanford, yours truly in the IGI, and many physician scientists and scientists across the nation and in Europe can design and trial preclinically a CRISPR for a given disorder of the immune system, and there's about 500 of them. But remarkably, I think the most advanced effort in that space is Matt's for X-linked severe combined immune deficiency. There are relatively few late-stage CRISPR efforts for a set of diseases that we could potentially edit today. So what do we do? The first thing we do is find ways to go into the clinic as soon as we can. And Matt and I have made this point in public many, many times, and I actually am very curious from Matt's perspective on all of this. We can only learn about clinical safety and efficacy from clinical trials. And it would mean a lot to the community, but also for us submitting INDs for Dr. Marks's review to say, well, this is from a platform perspective, this is what we've already done clinically, and this is what we have seen. We would like to reuse this platform component. The other is, I think, what will be essential is more or less why the raison d'etre for this summit, and thank you, JC, thank you, Vanessa, thank you, Sadek. This would require highly innovative private academia partnerships, where there's a lot of asset matching, sort of the 1 + 1 = 7. But finally, and I would love to hear Matt's perspective, I realize I'm sort of waving my own flag here. I really think that academic centers of clinical innovation should play a leading role in this regard. Again, think of what Bruce Levine and Carl June and Penn have done for CAR-T. And I think centers, for example, such as Stanford, where Matt leads the work and others, absolutely should be the place where this manufacturing innovation, regulatory innovation, and the unmet patient need could be the place where such enabling clinical trial data could see the light of day.
Let's pick up that one, you, Matt, and this goes for you first and then George. So we're talking about artificial or synthetic hematopoiesis.
I love that term.
That's right, that's right. Of course, George was talking before about almost with 20,000 edits, you know, to redefine a lineage if you want to. I mean, again, if you have to. So what is, having bone marrow transplantation being a radical cure, and we have known this even in autoimmune disease for many years, but of course one that comes with a lot of consequences. What is the future of synthetic or artificial hematopoiesis or cell lineage modification?
Well, I'm going to say the future is bright. What else am I going to say?
Yeah, that's right.
But I'll tell you why. But I think the path there is that first synthetic hematopoiesis has to be proven to be safe and effective when you're correcting an underlying genetic disease-causing mutation. That's the proof of concept. That's going to establish the platform. And then I think you then can build in, you know, again, I referenced this earlier this morning, is engineering the hematopoietic system to deliver proteins that maybe they don't normally deliver. That's our first step. And so I'm okay with the iterative nature of you build from correction to delivering a single protein to maybe delivering a single protein coupled with a resistance marker and building up so you really create more and more of a hematopoietic system that doesn't look like the original hematopoietic system. That being said, that has to be coupled with manufacturing changes and safety assays that as you layer on these changes and go farther and farther away from what nature tells us is safe, how do you preclinically evaluate that this is likely to be safe as you do this in a human? And again, a concept that everyone knows, it probably is done in diseases where there's very, very high morbidity and mortality. And so the risk-benefit says, let's try it in these diseases, and then if it's shown to be safe and effective, the needle moves, and you apply the same concepts to less severe diseases. I'm going to just respond to Peter's comment about manufacturing. I love the term that manufacturing changes that are substantially similar can be implemented relatively quickly. I love it because it's vague, but I also hate it because it's vague. And you can get some uncertainty about whether the change we make is substantially similar, and therefore I'm not going to have to go back to ground zero, or is the change going to be perceived as different enough that I have to go back to ground zero? And I think a discussion about what substantially similar means as you're talking about an ex vivo manufactured cell, probably we need to get more granular about that.
Peter, just because Matt mentioned this, do you see a trend in which, because medicines are much more personalized, safety, of course, patient safety will continue to be paramount, but not population safety, but rather manufacturing CMC control and establishment of, you know, very strong processes that are super regulated, that are super clear, and because it's going to be in 10 patients or in 100 patients versus 100,000, a more emphasis is put on CMC reg versus population safety or not?
I think actually that this goes back. Yes, I do think that's right, but it's actually quite funny. It's like it's a little bit of like a return to basics, because this is where biologics got its start, right? Where the process is the product.
That's right.
And the quality baked in from the start. So I think if we have a high quality, I think for many of these things, right, if we believe we have a high quality product in terms of the way it's manufactured, I think given what we know about these classes of products, we'll feel more comfortable on the individuals seeing these used, right? In other words, products that have good, that we know have a high degree of purity from adventitious agents or contaminating DNA, et cetera, et cetera. I think this gets us back to very basic biologic manufacturing, much as it was at the turn of the century. That is, the turn of the 19th to the 20th centuries.
Right. George, the way that you did your presentation, you were going through different layers of this convergence, which was very exciting to us. As you look at the future of medicine, do you envision now that, for example, the same way that you dealt with, and the team dealt with porcine retroviruses, that certain endogenous retroviruses for us that get de-repressed when cancer gets activated and we learn from them with time, is something that we might do? What are the different layers of that genomic medicine that you see in the future for us?
Well, sticking with hematopoietic stem cells for a second, they're particularly interesting because they're one of the most common transplants done today. They also distributed, the progeny cell are distributed throughout the body, so they can impact multiple tissues. And I think we're going to increasingly see sort of enhanced cell therapies. They're not just doing a particular job. They might be in there for a lifetime. And so you want them to be senescence resistant, pathogen resistant, cancer resistant. And we know how to do these in animals. We need to do it in a way that's safe. And it can't just be preventative medicine. As much as I love preventative medicine, it's a little bit harder to get it approved, rightly so, because you start out with somebody that's healthy. So what I think we're going to see more of is dual use, where you have, it gets first tested in the high morbidity, as Matt said, cases, but then it can be used and tested at our leisure on the more preventative scales. After we get a lot of familiarity with it for the high morbidity cases, and I think that age-related diseases are going to be the classic ones, because so many people will be able to defray the high costs of the R&D and the clinical trials.
Carl, just continuing with this theme of progressive, but rapidly progressive due through iteration in the clinic. I mean, we just heard it several times today. The way to advance genomic medicines fast is to iterate in the clinic with safety, with warranties, with manufacturing, but that's how we learn. And I heard you a number of times to say, well, one could envision a world without chemotherapy. So how far are we, and how does this relate to the conversation that we just had about bone marrow transplantation, for example?
Yeah, you mean how can we avoid using chemotherapy?
That's right.
Yeah, I think there's very promising approaches now with either cell-based therapies or ADCs, to do myeloablasive approaches and, surgical removals of certain lineages of cells. And so I think that's going to be a next approach that's going to follow on the heels of hopefully successful treatment of AML, which is like a stem cell disease. And I think that's going to enable, myeloablation that's not chemotherapy based, as a hope I have at least. I think, another major area that's going to happen and that's going to impact what Peter does on a daily basis is, these recent reports of the use of CAR T cells in lupus. I think we're going to see a lot of approaches now using engineered cells for autoimmune disease and organ transplantation. The Holy Grail has been, can we do an organ transplant or even a xenotransplant and not have to give, systemic immunosuppression? I think that's eminently doable. It's an issue of how you bring it into the clinic and and so on. But I think it can surf on the success of cancer therapies, and that's going to, like remember the first bone marrow transplant for HIV that cured Tim Brown. That was because he had leukemia, and we found out you could actually get, in some cases, sterile elimination of a virus. So I think, we're going to see this year many approaches now in autoimmune disease, and we've talked about it for years. Finally now, I think we have enough safety with genetically engineered cells that this is going to become a reality.
Right. I think this, maybe Matt and Fyodor, this intersection between precise gene editing or gene modification and cell therapy and selective cell therapy, ex vivo or in vivo, however it's done, I think could put us on a faster track to get a large impact to patients. Is that true? Please.
There's this saying that a pessimist is a well-informed optimist. So being a well-informed optimist, I think a legitimate threat to this vision is actually what Matt said this morning, which is that we have a tremendous amount of very healthy progress across various biotech companies that are treating disease 1 versus disease 7 versus disease 25. And the 64,000 base pair question is, how does all this momentum become the proverbial rising tide that lifts all boats? And one approach is the following. Berkeley had this wonderful slide this morning in which he talked about, quote, a technology stack for accelerating genomic medicine. And Vanessa spoke about, quote, unlocking 7,000 indications. I think that, and Peter, thank you for your vision in leading the gene therapy consortium and saying, look, AAV9 is AAV9 is AAV9, right? So the more such platform capabilities exist that populate that vertically integrated technology stack, wherever that platform capability resides, it could be in an academic center, in a for-profit company, then the sooner we can get to accessible and affordable personalized medicines, because we don't have to, reinvent wheels by redoing manufacturing or redoing some talks or redoing some other analysis. As I've said publicly, I'm happy to say again, I'm very grateful to Mark and his team at IDT for building RampSeq. It may seem like just a technology, but it's wonderful to just get it off the shelf and put it into your IND. So the more that we can populate the technology stack with such solutions, and the more we can put it into clinical development and see how it performs, the closer we'll come, I think, to that future of accelerated medicines on demand.
Yeah, I'll comment on the last. I think standardized assays, standardized analytics will be really important, and RampSeq is a contribution, and I think there's others. In terms of the impact of precision, I think it can impact in at least 3 ways. One is, of course, safety. I think the more precise you are, the more comfortable the entire community will feel about your drug product. The second is precision should give reproducibility, and that also is something that could expand the use. And finally, I think this is something that maybe people don't appreciate, and maybe I'll get some pushback. Precision gives purity. That is, your drug product or what you're doing is more homogeneous across the population, and therefore variability is, well, there's always going to be variability because of the human you're giving the drug product to, but at least the variability that comes from the drug product itself will be reduced. And so I think precision enhances things both from safety, reproducibility, and purity, which are key components of any drug product we want to think about.
Right. I mean, we were talking about critical product attributes. Peter was talking about going back to basics, everything that we have developed in terms of drug product and drug substance characterization for other modalities has to be developed here to make sure that we feel comfortable. But since you talk about this precision, there is another aspect of us that is very close to Danaher. Almost 50% of our business is a diagnostic business. And I think it was Ed, that one that said, well, let's not call it diagnostics, or maybe it was this. Let's call this, how do we measure during, of course, propensity, chances to respond, relapse or flare, or MRD, of course, to what extent precision diagnostics are critical for genomic medicines? Of course, at the moment, we just get a sequence, and of course, we heard in the previous panel, you get a sequence of the baby, you get a sequence of the tumor, and that enables certain things. But what other precision diagnostics are needed to enable this field? Maybe I start with you, Carl, and then we go in.
You know, there's, right now, we can't predict who is going to respond and who not. I mean, this is, we heard this morning, I think Phil Sharp said, you know, I mean, each cancer is an orphan disease. They're all unique. And so until we have that routine, we need both precision diagnosis of what the synthetic lethalities are to each cancer to design then, you know, effective therapies. And we don't have that now. And then we're going to also need to understand that the host immune system of the patient and to how best exploit that, or else we use off-the-shelf cells and so on. But right now, understanding each person's cancer it's a huge area. And then another is, I mean, these spatial omics, being able to look at biopsy specimens and figure out what resistance mechanisms happen to patients on trials. Unless we learn from that, I mean, we're just going to fail. So I think that's amazing. And the good thing now is there's been these huge advances with these spatial omics to be able to analyze people after therapies and understand why it hits the mark or misses.
Right. George, maybe you go after and then at Fyodor. So of all the technologies that, of course, you and your colleagues develop, a lot of them are dedicated or focus on therapeutics, but this concept of drug product characterization and analytics, or even the concept of precision diagnostics or precision monitoring of the drug or the response or the propensity, what are the most exciting things that you guys are doing or that you have seen in that field?
I mean, considering that the cost of many diagnostics, especially nucleic acid-based ones, have come down by 7 orders of magnitude, I think we're severely underdiagnosed. It would probably improve all of our clinical trials as well as phase 4. And we're getting some that might be both general purpose and wearable. I mean, we've already got sort of, 24/7 glucose monitoring, but just imagine that could do a more general set of small and macromolecules. And this, for example, the molecular transistors, Roswell and others, other sensors could be among that. We're not in any way limited. The revolution is not limited to nucleic acids. So I think that would be quite interesting to have continuous monitoring. That's far less risky, let us say, than not monitoring, and it's certainly less risky than applying therapeutics in general.
Yeah, good point. Fyodor?
George, I love the way you said it, that we're all underdiagnosed. And I think Exhibit 1A are the subjects on CRISPR gene editing trials. And just to give you an example, CRISPR Therapeutics, we all hope, together with Vertex, will have our first approval, fingers crossed, for sickle cell disease. It's a magnificent achievement. And they've released some data on efficacy, but let's just say that the public information available on all sorts of interesting biomarkers, both genomic and proteomic, on how their patients are doing is lacking. I'm sure the agency has seen it, but it's not in public release. Contrast that with a publication of 2 pediatric subjects from China, which have been gene edited using the exact same approach that CRISPR Therapeutics has taken. And they published, I think, a Nature paper in which there's 6 figures of clinical data from the patients. And professional gene editors such as Matt or myself are looking at those figures and going, oh, wow, so that worked well, but this could be tweaked. So I think a huge opportunity for precision diagnostics in our world of gene editing would be as much patient follow-up analytics as we can humanly gather, so we can learn from the lived experience of the patients how to treat that community better.
Peter, what do you think? I mean, the importance of continuous monitoring or semi-continuous monitoring, or at least, particularly in the field of genomic medicine, not only because of price pressure, but also because we want to make sure that we, in some cases, treat. There you go. I think we lost him. No, no, you're back. Because in this field is one of the ones that, as we said before, we have the opportunity to treat preventive, but not preventive blindly. Prevented with a mutation that gives you a significant chance to go. So how prepared are we from the regulatory perspective to deal with that?
Yeah, I think this is not, this to me, I think we're reasonably well prepared for dealing with this issue. It's something we've, I think we're lucky that my colleagues in the Center for Drug Evaluation and Research and the Oncology Center have been used to this concept. I think we'll be able to keep this. I think we can keep up with.
Excellent. Matt, in the areas that you work, how is precision medicine and precision diagnostics? How do you project the future of this? I'm going
to be a good lawyer and not answer a good politician and not answer your question. Beautiful. And rephrase it that sensitive diagnostics is related to precision.
Sure.
And I love the term brought up by an earlier panelist about temporal accessibility, and treating diseases earlier in the course is always better. And so precision, sensitive, predictive diagnostics will allow us to identify the patients as early as possible who deserve the therapy that might transform their lives.
Yeah. I think, we wanted to finish with this topic on purpose because we talk a lot about manufacturing and, of course, the importance of driving manufacturing costs down. And obviously, this is one of the areas where we really can help and we will help. I think there is this proliferation of therapeutic platforms that is still, of course, biology is complicated. We were reminded a number of times we're at the infancy, and then not at the infancy, but still, it will be complex. But the one area that this field has that we must focus on as well is to make sure that we identify the patients early, that we monitor the response, that if they don't respond, they get off therapy, of course, or we go to additional therapies. We were talking before about several therapies that, of course, at this point are unthinkable from the financial perspective, but eventually they will be. And we see, for example, in Duchenne, how you want to delay treatment with a virus with an ASO as long as possible because you want this mass being built before you transduce. So I think that component of precision, or how is it, sensitive diagnostics that have a temporal deployment through not only your disease, but your health, your predisposed health is important to us. So listen, we didn't take any questions from the audience because we know that you guys are going to jump at the speakers at the next coffee break or at the cocktail. And we want to thank our virtual participants. Thank you so much. We know that sometimes it's more difficult to be online, but it was great to have you. And of course, to our panelists here. Thank you so much.